The overall goal of this methodology is to measure the uptake of glucose and its transporter on different monocyte subpopulations.
This method can be used to answer key questions in the immunology field, such as what is the importance of immune cell metabolism in disease outcomes? The main advantages of these techniques are it is quick, easy to perform, requires no radioactive material, and requires only a small volume of blood.
Within 1 hour of collecting the blood samples in citrate ACDB anticoagulant tubes, place the samples into a biological safety cabinet. Next, add 100 microliters of blood from each sample into individual polypropylene tubes and treat the cells with 2 milliliters of 1X lysing solution on ice, pipetting gently to mix. After 15 minutes, centrifuge the cells and follow this with 2 more spins in wash buffer. After the second wash, use a pipette to carefully remove as much of the supernatants as possible and resuspend the pellets in 100 microliters of wash buffer on ice. To identify the specific monocyte subpopulations, stain the cells with 5 microliters of the appropriate antibodies per 100 microliters of cells for 30 minutes on ice in the dark. At the end of the incubation, wash the samples 2 times in wash solution and fix the cells with 200 microliters of 0.5% formaldehyde in PBS. Then, analyze the cells within 24 hours.